Let's take a brief look at what you're doing this week. We are in week 2, and you have 2 major things that you're looking at. One is that you're going to set up an experiment on population growth in duckweed, and then you're going to be measuring that population growth over the next few weeks. The second assignment that which you will complete this week is the species accumulation curve. So let's go into Module 2 and take a look. So you have your weekly objectives and the learning activities. In your learning materials, you have the weekly lecture. You also have some information about field and lab safety, so make sure you take a look at those before you do your experiments. You also have resources here on how to identify birds because you're going to use that for your species accumulation curves. Your first assignment this week is to set up this experiment. It's a gallery discussion, and you're going to set up your experiment on duckweed. And in this experiment, we're going to be looking at population growth. You have a video here that shows you what it looks like when this population grows. You also see individual plants here. And what you're submitting in week 2 is the setup of your experiment. You will put in your picture of your setup of the experiment and your hypothesis, and you'll put that into this week 2 discussion. You have instructions for how you're going to set this up, where you can get your duckweed, where you can get the Miracle-Gro to use, how to set it up. You'll be creating these 3 different solutions. You're going to use distilled water and Miracle-Gro, and it shows you the different amounts that you're going to put in. So you're going to have one that has a fairly high concentration of the Miracle-Gro, a slightly lower concentration, and then an even lower concentration. And then you'll also have one set up where you're just using distilled water. So these are your treatment. You'll have 2 cups for each treatment. So you'll have 2 cups that have just water, 2 cups that have the solution A, 2 cups with solution B, 2 cups with solution C, and each of those you're going to put 10 of these little duckweed fronds. You're going to count the number of duckweed fronds you see every day for 10 days. You're going to put that data into this file here. It's a Google folder. What you're going to do is to take that file, you're going to download it. When you download it, then you also want to make a copy and save that copy onto your computer. So that way you also can edit it. What you'll see here is you have days 1 through 10. You have the number of individuals that you'll have in these different conditions. So treatment 1 was the pure water. You have cup 1 and cup 2. Then you have 2 cups with solution A and all of your different ones there. So you'll put all of the data in there. I'll provide another video later that'll show you how to analyze these data. Now this is a gallery discussion. So in a gallery discussion, you're not required to reply to one another, but you are encouraged to do that, or at least encouraged to look at what others are doing. So you'll do your setup of your experiment. Your next assignment this week is to do a species accumulation on birds. It shows an example of a species accumulation curve. And basically what it means is if you go out and you start sampling, if you only sample a small number of locations, so let's say you have a big field of wildflowers, but you only sample a very small portion of those, then you're going to get just a few species. And take another sample and then another sample and another sample. The more samples you take, the more new species you'll acquire. When you take 2 samples, if you have 2 of the same species, you don't count those. You only count the new species for each time here. But every time you increase your sample size, you'll probably get some more species. But at some point, that's going to start to level off. Because at some point, if you've sampled a lot, you're going to just be resampling the same species. And that's what happens when it gets flat up here. You're sampling over and over again, but you're not getting any more new species because essentially you've sampled intensely enough to get them all. When you're looking at a study and you want to know how much do I really need to sample, you can look at these curves to decide whether you think you've got a good enough sample that's an accurate representation of all the species that are there. These curves here that continue to go up, we haven't sampled enough yet because it's still going up. But when they flattened off, we have sampled enough. So this is looking at those species accumulation curves. So whenever you're going into an area and want to estimate species diversity, it's important to take a look at these species accumulation curves to get an idea for what kind of sample size you're going to need. So it's asking you to go out and find an area where there's a bird feeder, or maybe there's an area where birds congregate near you. Maybe it isn't a bird feeder, maybe it's a pond or something. So if you can find an area where you're going to see a variety of different birds, you want to use that area. So if you have a bird feeder in your yard, you can use that. You can use other areas. Again, some park areas will have a lot of species that come to them. Maybe not so much because there's a feeder, but because there's ample other resources there. If you really can't get out, there are a couple of different webcams that you could use for this, but it's a lot more fun to get out and look at them in real life. So what you're going to do is you're going to watch this area for 2-minute increments. So over this 2-minute period, how many species do you see? And then you're going to do it again. Over the next 2-minute period, how many species do you see? And then of those 2 samples, how many were new species? So when we take our 2 samples, we don't want to add the overlap of species. We only want to include now the new ones. In this data table here, your first sample, this is your first 2-minute period. How many species did you observe? Well, of course, the first sample, they're all new. But in the second one, you're going to see how many total did you observe, and then subtract out any overlap that you had, only the number of new ones. And then over here, you're going to look at the cumulative number. So if I had 5 here in my first sample, and then 3 in my second sample, my cumulative number over here is now 8, because I have a total of 8 that I've seen so far. So what you want to do then is to create a graph that looks like this. So you're going to have the number of samples, these are your 2-minute time periods, and the total number of species richness. And you're going to try to see where this graph that you're creating levels off. As it says here, you want to continue sampling until it's leveled off for at least 3 samples. So if you're still finding new species, then you want to keep going until you get essentially 3 samples in a row where you're not finding any new species. And then you have some questions that you can answer about what you're finding and about the process here at the end. After you've collected your species accumulation data, if you notice here in the announcements, we have a video for how to create the graph in Excel for your species accumulation curve. So if you click on here, you'll see a video that describes to you how to make that graph. As always, if you have any questions in the assignment, make sure you contact me. I can either answer you in an email or we can agree to meet virtually. As always, also make sure that you look at the grading rubric below the assignment to make sure that you are including all of the items that are required. So you can see the grading rubric and the number of points. So you want to always go back and double check the instructions to make sure you have everything and the grading rubric.